organisms, all living things, especially microorganisms, which are very, very small, cannot showing by naked eye, and you have to observe these microorganisms by microscope. First of all, also, there are two types of cells, eukaryotic cells and prokaryotic cells. The bacteria belong to the prokaryotic cells, very, very simple. It represents two domains of organisms, bacteria and archaea, all blue-green algae. Bacteria are the most abundant diversity organism on the world. Eukaryotic cell, especially for human and animals, much longer, more complex, have all the constituent, but in advanced stage than prokaryotics. Prokaryotics, especially bacteria. Bacterial pathogens, you hope to know that there is about 2,000 different microbes all over the world causing the disease, pathogenic microorganisms. About 10 million or 10 billion new infection every year all over the world, including human, animal, birds, from which about 12 million diseases per year. The zoonotic disease, about 3 to 4 million diseases per year. The microorganisms, or the bacteria, living mostly as a parasitics to another host, causing the damage of this host. We hope to know about Antony van Leeuwenhoek, the first German scientist which discovered the microscope. About 1632 to 1723. First discovering which dealing with the world of bacteria. Then John Tyndall, we know about Tyndallizations, which discovers the sterilizations. And Ferdinand Cohn, which talk about spore and heat resist form of bacteria. Another contributors all زي ما بنقول كده الملهمين لعلم البكتيريولوجي لويس باستير and روبرت كوخ. Louis Pasteur showed the microbe in some food poisoning or spoiled food materials and developed the pasteurizations and developed a germ theory which dealing with the detect the pathogens and observe the disease. Second contributors, Robert Koch, the father of bacteriology, who mentioned the Koch's postulates, and we later discussed it in details, which composed of sequence of very steps experimentally to verify the germ theory. Who also discovers anthrax to be the cholera and developed the culture method. He worked first in the Pasteur Institute in biochemical and staining lab. And then we discover all these diseases. Bacteria, first step, it's taxonomy or classifications, which facilitate or aids us or help to identification of these pathogens, which beginning by Carl von Linné, who discovered the taxonomy of the microorganism. Nomenclatures, it names a specific or systemic name of bacteria. First one related to the genus of the bacteria and in capitalized form. The second mean species by lowercase, and both are italic or underlined as Staphylococcus aureus. Staph, it means the species, genus, Staphylococci, and aureus meaning the species. Also Escherichia, genus Escherichia, coli, it is a species of Escherichia. Also, you have to know nomenclatures. It means the number or code of the microorganism, as ATCC, as we mentioned, or hearing from some literatures or authors about some microorganism named by ATCC and the number. This nomenclatures classification. Now, the microbiology, the science dealing with the microorganisms. Bacteriology, science dealing with bacteria. It is a eukaryotic cell. All component of the bacterial cell, as we know, bacterial cell wall, cell membrane, ribosomes, and the capsule, flagella, endospore formed our plasmid. Bacteria multiplication by binary visions. Binary vision, it means 2, 4, 8, 16, 32, 64, etc., etc. Now, this is the structure of the bacterial cell. Capsule or slim layer in some bacterial species, followed by cell wall from outside to inside, and then the cell membrane. Cytoplasm have the component of ribosomes, nuclear bodies, and then the flagella and the plasmid, which carry some chromosomes and genetics composition of the bacterial cell. Now, classification and its identifications based only on morphology and nutritional requirements, biochemical reactions, and biological activities. First, morphological classification. The two forms, filamentous form as in Nocardia or Actinomycetes with Streptomycetes, or true bacteria like most gram-positive and gram-negative bacteria, it is true bacteria. Now the shapes of bacteria, either cocci and its gram reaction, gram-positive or gram-negative microorganisms, were arranged in cluster or chain or diplo or single, or arranged in pair like gram-negative row cocci. Rods or bacilli or short bacilli, gram-positive or gram-negative. And the third one, accordingly staining the Ziehl-Neelsen acid-fast microorganism, either by staining the Ziehl-Neelsen or other. Other form like vibrio, curved rod, comma-shaped microorganism, or spiral form like Keats, which is spiral filamentous. This is the shape of the bacteria, cocci, bacilli. Now this arrangement, it is very important in its classifications, like the cocci or single, chain like microorganism, grapes like, or clusters like Sarcenia microorganism. And the form of arranged of bacilli, either coccobacilli or bacilli, or diplobacilli, or a chain millistriped bacilli. Now, this is a strategies for for bacterial identifications, include morphology and staining reaction, culture characters, biochemical reactions, antigenic characters, serology, antibiotic susceptibility, animal pathogenicity and toxogenicity, and last, molecular typing. All these strategical steps must be followed for each microorganism to reach to its complete identification. Now, the close curve of each bacteria, which begin with a lag phase. It is a phase for growth only. Then followed by logarithmic phase, which includes the multiplication and the increase in the number of the bacteria. And then stationary phase. Stationary phase, it means the number of new bacteria equal to the number of this bacteria. And this فترة يسموها فترة ثبات في العدد البكتيري. Stationary phase. And then decline with this phase, more bacteria did new ones. This is about 24 hours bacterial growth curve of bacteria. Growth condition is very important. To offer these requirements when you want to culture any bacteria and to identify it, you have to offer these requirements, including in temperatures, which are optimum for each microorganism, pH, which differs mostly from 6.5 to 7.5, but some microorganisms like Vibrio cholera, it needs acidic pH like Helicobacter pylori survive in acidic form. We have this knowledge before want to culture any microorganism or prepare the specific and most suitable media. Also, sodium chloride concentration should be about 0.9%. هو الفسيولوجيكال ساين العادي. لكن ملاحظة إن بعض البكتيريا زي الستافيلوكوكس أوريس الإبيديرميديس يعيش على السكين في الأسيدك، طبعا عارفين السكين سويتي، فممكن تعيش ب 10% من الصوديوم كلورايد وبتفضل عايشة. مفروض أعرف كل الريكوايرمنتس بتاع كل ميكروب عشان توفر كل احتياط إمكانيات اللي محتاج لها. Nutritions, some bacteria need iron, sulfur, hydrogen, nitrogen, source of carbons, also osmotic pressures, also anaerobic condition, it needs oxygen or not according to the type of the bacteria. Now, the gram staining. And after Christian Gram, which discovers this stain, can differentiate the bacteria into gram-positive or gram-negative. Gram-positive takes the purple or bluish color, and the gram-negative takes pink or red color staining. Gram state principle to determine the gram state, either gram-positive or negative, according to the composition of the cell wall. It first begins with the staining with crystal violet or primary stain for staining the cell wall, and then iodine as a moderate, and the last for carbofuchsin or sofranin as counter stain. This is a principle. Gram-positive bacteria retains the color which primary stain of crystal violet نتيجة وجود البيبتيد جلايكوجين في السيل وول بتاع الجرام بوزيتيف بكتيريا، في حين وجود اللايبوبوليساكرايد ما بتقدرش كانت retain or keep the primary culture its cell wall, and by decolorization with ethanol or alcohol it lose this primary culture and then stained with the counter stain like carbofuchsin, so it takes the red color. Now these steps: crystal violet, primary stain, dye cell wall, then gram iodine fixes the primary stain, and transition with ethanol, which in gram positive retain this stain and they give gram positive, or gram negative, which lose this stain and become colored by the counter stain of carbofuchsin or sofranin and takes the red color. Now we can see the bacterial cell in gram positive and the gram negative. So the gram positive, it is very important, but peptidoglycogen in cell wall of the bacteria. So it takes the primary and can't lose this primary stain. But small band in the gram negative, and it instead of it, there is a large area from the lipopolysaccharides, periplasmic with lipopolysaccharide, which lose the primary culture and attack the secondary one, which carbulfoxin. So it takes the red color. Now, if gram-positive and gram-negative bacteria, all steps it's equal. The decolorizations in gram-positive, the cell wall retain بيحتفظ بالصبغة في السيل وول، في حين decolonization في الجرام-نيجاتيف نتيجة وجود البيبولوسكار بيفقد الصبغة الأولية، وبالتالي لما يجي ياخد السفرانيل أو الكربولفوكسين ياخد الريد كلر. ده السفرانيل أو الكريستال الكربولفوكسين بتنزل مع الواشنج ويفضل محتفظ بالبرايمري في حالة الجرام-بوزيتيف. This is bacterial cell. It's a difference between constituent gram-positive and gram-negative bacteria. And this reaction of gram-positive bacteria or cocci, blue color or purple color, and this pink or red gram-negative microorganisms. Now, we say about this staining. Now, the acid-fast staining. Some microorganisms have a more and high amount of lipid content in its cell wall and wax in their cell wall. So it can't staining with the primary stain like carbulfoxin or crystal violet. So it needs some modifications, which in acid-fast staining to stain the microorganism like mycobacteria. It takes the shape of Mycobacterium bacilli and its red color by Ziel-Nelson. Acid-fast staining method, either Ziel-Nelson, it is a hot stain, it needs heating, or canyon stain, it's called the method, or modification for each of them. Now, the culture media types. We have to know there is many types of media. You have to know at first what you want to do. Some microorganisms it need enriched growth, like Bifidobacterus, like Salmonella. And then you want to selective culture to choose the selected microorganism or definite or detected microorganism which you need to identify, like Salmonella. You want a selective culture media like SS or Methylobuluf in case of E. coli. It is a characteristic culture, characteristic features on the surface selective media. But in enriched media, it needs some fluid media, liquid like broth or some cases antibiotics, broth and in numbers, and allow it to growing on the selective culture media. Some media, culture media, it needs antibiotics in case of nocardia. It is Taylor Martin agar. Or indicator media like McConkey agar, which contain bile salt. It's very essential to inhibit the gram-positive bacteria and initiate the growth of Enterobacteriaceae or gram-negative rods like Salmonella or E. coli, or as McConkey agar. So it is very essential to know what you want the type to prepare the type of culture media, which is suitable for your microorganisms. Also, like in case of Pasteurella, you have to know the Pasteurella maltosida gram on blood agar, but not good in McConkey agar. Then Mannheimer hemolytica or Pasteurella hemolytica previously, it causes hemolysis on blood agar and it causes indole positive. Correct differentiation on the culture media and the differentiation by chemical reaction to reach its identifications. When you, in case of pneumonic coughs or any pneumonic or respiratory manifestation and you suspect Pasteurella, when you have the culture of Pasteurella, you know it growing on blood agar or not, growing on McConkey. The Pasteurella hemolytica causing no death for mice, still alive. But Pasteurella maltosida is caused this of the injected or inoculated mice in 17 to 24 hours. This dead mice and take from the heart blood, and you can stain it by the Ramme stain.
بس بنعتذر فيه كده يعني الإنترنت فيه مشكلة بسيطة وهنرجع لكم على طول. يعني ثواني وهنرجع لكم.
Now, about the bacteria, which bacterial pathogenesis and its impact on the animal or human being. There is a relationship between the bacteria and the host. It depends on the two items, very important. The virulence of the parasite of the bacteria itself, and second, the resistance of the host or its immune status, and equal the state finally, if diseased or immune or resist infections. Mostly it is very complex relationship between the host and the bacteria. Some microorganisms, commensals, like Aeromonas in the intestinal intestine of the fish, but in some cases cause septicemia in immune suppressed fish. In some cases convert to pathogenic microorganism. There is two ways for actions. First of all, direct damage to the bacterial cell, to the host cell, and second, stimulate the host inflammatory response or immune response, and finally damage this host cell indirectly. Pathogenicity, it means the transmissibility والcommunicability، التواصل والتواجد ما بين البكتيريا والريسيفوار او الفريش هوست. نمرة 2 السرفايفل في النيو هوست، لازم يعيش. نمرة 3 انفكتيفيتي، يعني يعمل العدوى، وابيليتي انه يوصل يعدي resist الديفنس بتاع الهوست by its virulence. This the issue. First of all, it needs tools for transmissible and to attack the host cell. Second, to survive and infective the host cell. Now, it is a virulence of microorganism. It's a capacity of the bacteria to induce the infection and cause the disease in the host. And this virulence facilitated by some component of the bacteria, all of which each part of or constituent of the bacterial cell play an important role for the virulence or disease process, as we know now. Virulence factors, molecule component في bacterial pathogens, had the ability to cause the disease. First of them cause damage to the host through production of toxins, and as mentioned, that indirectly damage and promote colonizations and attack and survive and infective the bacterial the host cell. All these steps very important in sequencing. First of all, entry the host. Second, adhesion of the bacteria to the cell wall. Then invasion or propagation of the organism inside the bacterial, inside the host cell. For the damage which induced by toxins or inflammatory response. And the second is how to overcome the host secondary defenses of the host. All these processes and the steps are very important in orders. First of all, microorganisms can enter the host cell through the respiratory air passage or gastrointestinal tract, urogenital, skin, or cut or burned or puncture the skin. Just enter the pathogen inside the host cell. First of all, the bacteria must be overcome the host defense like phagocytosis or acidic environment of skin or saliva or stomach content or urogenital tract. And then some enzymes formed in saliva and stomach and the small intestine, which maybe compete the bacteria. Second of all, the bacteria have to adhere to the cell wall of the host, either by flagella or villi like group Streptococci, or through adhesion molecule interaction between a specific receptor في mammalian or host cell and the receptor of the bacterial cell surface to adhere to this cell. And this adherence were enhanced by the adhesin production of the bacteria. تفرز مادة زي الادهيسين تساعدها على الالتصاق بالهوست سيل. إذا الادهيرنس ده enhance الفيرولنس طبعا by preventing البكتيريا من ممكن البكتيريا بالبريستالتيك موفمنت بتاعت الانتستين او الكومبوننت بتاعت السلايفا او اليورين انها تموت البكتيريا او تكمبيت البكتيريا. لكن هنا الادهيرنس بيمنعها من هذا، ودي تولز مهمه جدا للبكتيريا انها تقاوم وهي داخله مقاومه الجسم. الادهيرنس two common strategies فامبريا and monomeric بروتين ادهيسين. الادهيسين بيساعد في الالتصاق او من خلال الفامبريا الموجوده او البيلاي usually carbohydrate in nature 8 في production can be blocked in some cases by antibodies which migrate to the site of infection, and sometimes can the bacteria overcome and combat the antibody reactions. Or the adherence occurs through monomeric protein adhesin mediated by cell surface protein of bacteria and bind the host cell and attach it or adhere to the host cell. Like here, there's a host cell and this bacteria either through receptors like this, and this receptor for the fimbriae or villi, or through the villi or flagella directly and penetrate the host cell through penetrated factors or infector factors produced by the bacteria. Now, after adherence, the bacteria have to invasiveness inside the bacterial cell. تخش جوه البكتيريال cell وتستغلها consumed its component nutrition for itself. To invasiveness successful is facilitated by several enzymes, coagulinase like in Staph aureus, hydrogenase, which liquefies the tissue and then facilitates the invasion of microbacteria of any bacteria inside the host cell. Also, bacterial invasion enhanced by motility through flagella or chemotaxic induced swimming using some nutritions. And finally, invasion of host cell becomes the bacteria is intracellular pathogens. The two-mediated invasion, after invasion, to establish inside the host cell, either by zebraing the present ligand on the surface and compound or binding to FC portions of antibody or CR portion of the complement in the blood to compensate or to compete the phagocytosis and alive or survive inside the host cell. All other items by triggering, it means the bacteria inject effectors help in the invasion inside the cell, like case of Salmonella, and after adherence and to the bacterial host cell, and then in penetrating inside the bacterial cell, the host cell. Then it began to produce its toxins, either endotoxin or exotoxins. Endotoxin, it means the lysis of the bacterial cell and liberated of the its toxin inside the host cell, causing its damage. Or exotoxin, it is a bacteria, it is alive and intact, bacteria سليمة ولكن diffuse its exotoxin outside the bacterial cell, like في حالات الستاف اوريوس. البكتيريا بتبقى سليمة لكن الإندوتوكسين جوه البكتيريا have to lysis and liberated its toxin inside the host cell. Some types من oxytoxins which ingestion first by toxin like the botulism, Clostridia, or colonization on the surface followed by toxin production like cholera or diphtheria toxin, or exotoxin produce bacteria in tissue and it's spread during its like Clostridium perfringens toxin. Different tools for toxin in cause damage. As we mentioned, diphtheria toxin, cholera toxin, Shiga toxin, or the Clostridium toxins. The bacteria, for its invasion, needs some facilitator like factors or penetrating factors. Just for direct penetration, which help its invasion, would if present small cracks of the skin, small wound, animal bite or arthropod bite, all this facilitate the invasion inside the bacterial cell. Then the bacteria have to multiply, and we remember the growth rate of bacteria to increase in size and to begin to multiply, first growth and second multiplication, to increase in number to compete the phagocytosis or the immune system components of the host. Then the bacteria have to spread inside the body, inside the host. Sometimes it localizes the infection like gonorrhea or salmonellosis. The bacteria زي السالمونيلا في الانتستين تعمل إسهال. It is localized في الدايجستين. ولكن في بعض الأوقات systemic infection like الإنفلونزا or meningitis, pneumococci, it is a generalized infection or systemic infection called system. Or sometimes begin like typhoid fever, localized and begin followed by systemic infection. The spreading here by three ways inside the host body: through lymph circulation or blood circulation or a nervous system. Each microorganism, each bacteria have its own infective dose. بتختلف جرعات الانفكشن من ميكروب لميكروب. Like ميكروب زي السل, TB, one bacilli can infect animal or human and cause infection. In case of Staph aureus, you have to know that 3 multiply in 10 أس 8 microorganism to begin to produce toxin and cause infection. This depend on the character of the bacteria itself and also behavior or host immune defense. How the bacteria avoid the defense mechanism? ازاي البكتيريا بقى بتواجه الجسم بيطلعه عشان يصد العدوى. معلش انا بستعجل شويه عشان ايه اقدر اكتر معلومات نوصل لها. First of all, الكبسولة and its nature to keep the bacteria, protect opsonization from phagocytosis, also help in its adhesion and in penetration and invasion in the bacterial cell. قلنا في الأول خالص إن كل كومبوننت في البكتيريا مهم جدا في إحداث العدوى، وإن البكتيريا تكمل. Okay, and lastly, agricine. It is a substance below the bacterial cell, pathogenic bacteria, to enhance the virulence of the bacteria and cause paralysis للديفنس mechanism through production of its component urease, metaprotease أو mucinase أو hydrogenase enzymes. Also some enzymes like coagulase, as we mentioned, coagulinase or DNase, all these enzymes facilitate the protection of the bacteria from the immune system and phagocytosis. Then bacteria begin to obtaining its nutrient from the host cell, from the host cytoplasm. It is very nourishment material, and the bacteria consume the host cell for itself. Second, the evasions or how to overcome the immune system through serum resistance and the paralysis للmechanism of complement, or through capsule mediated resistant complement preventing من C3 و 3BH complex form up of the complement to complete its reactions, or through lipopolysaccharide of the bacteria which endotoxin bind complement items, molecules. Also, S layer of some microorganisms as Aeromonas encoded and Sula prevent phagocytosis and the action of antibodies. Resist of opsonization and compete the action of macrophages through capsules, and also prevent antibody-mediated opsonization and phagocytosis by masking. البكتيريا بتفرز بعض مواد زي الهيالورونيك اسيد تشبه المواد اللي موجوده في السيرم بتاع الهوست سيل، فبتعمل ماسكينج، بتحير الانتي بودي، مبقاش عارف هو ده غريب ولا من جوه الهوست سيل، وبالتالي الماسكينج ده بيمنع الانتي بودي رياكشن على البكتيريا. زي ما قلنا الاس لاير واللايبوبوليساكرايد بتفرز بعض البكتيريا بروتيز which compete الاي جي جي، زي ما احنا عارفين قلنا بيبقى فيه اذرع للانتي بودي مولكيولز، فبتعمل ماسكينج للريسبتور انها تعمل تشينج للارينجمنت بتاعها، فبتلخبط الاي جي جي فما بيقدرش يقاوم. او تعمل سويتشنج للانتيجينيك زي ما قلنا، او ماسكينج عن طريق افرازات معينه تعمل كوتنج للبكتيريال سيل تخليه اسكيب من الاي جي جي. Also biofilm reaction, which is very characteristic production of materials for drug resistance. It is very important to resist the antibiotic therapy. This slim layer slows the growth rate of the bacteria through biofilm production, or decreases diffusion of antibiotics through the bacteria, and the accumulation of the enzyme then contribute to resistance. Some enzyme, as we mentioned before, aggregate and accumulate to prevent the action of antibiotics. Then the resistance of present antibiotic-regulated phenotypes لsome bacteria, it means it's persistors. Some bacteria بيتسمى persistors اللي هي خلاص have the ability to resist the action of antibiotic, mostly in the E. coli, Pseudomonas, and the Staph aureus, and the Mycobacterium, multi-drug resistant strains through this phenomena of the bacteria. Now, اللي عايزين نقوله ببساطة إن البكتيريال باثوجينز highly adapted، متخصصة جدا، highly specialized. الانفكشن ما بيتمش ثرو كومبوننت واحد او اثنين، ده عباره عن بيسموه اوركسترا كامل للبكتيريا اتس كومبوننت تو انديوس ذا انفكشن. الكبسوله والتوكسيدز والسبيريشال ميكانيزم اوف ذا بكتيريا كان انفيد اند سرفايف اند بروديوس اتس harmful effect on the host cell. مهم جدا we have to know about the genetics which related to the mutation of the microbacteria, of the pathogenic bacteria, and phenotypic variations which occur from time to time to change the H antigenic characters, is genetic transfer of the bacteria and genetic basis for antibiotic resistance. All these items very important related to the bacteria itself. Now, last point I want to know, it's as a bacteriologist, what is the Koch's postulates? Koch's postulates, it means the microorganism which you represent in any cases and isolate in pure form. This microorganism has a character, and when injected again in the another microorganism, in the other host, it causes the same symptom and the same lesion of the disease. Then you can re-isolate this microorganism in pure culture from the new host, even laboratory animal, produce similar diseases, and it re-isolate it in a pure form again of any bacteria. Complete these items, it is related to the Koch's postulates. When isolate bacteria, you have to confirm, identify, and classify it, and prove that it is the main bacteria which cause the disease. Koch's phenomena, it is very important to differentiate between Koch's postulate and the Koch phenomena. Koch's phenomena, it is a hypersensitivity to Mycobacterium or tuberculosis infections. When Guinea pigs is infected and already infected with tuberculosis, and the challenge again with tubercle bacilli developed and exaggerated the inflammatory response, it means the Koch's phenomena. Not blocking the infection, not disappear, but it is a severe reaction on Guinea pigs after reinfection. Antibiotic sensitivity test, now it is a challenge، تحدي كبير جدا لوجود highly multi-drug resistant strains في كل حاجة في عالم الحيوان والإنسان. فمهم جدا انك تعمل antibiotic sensitivity صح عن طريق الديسك ديفيوجن ميثود، تعمل ستاندردايزيشن صح للبكتيريا الاونيكولم عن طريق 3 في 10 أس 8 او انبوبه رقم نص في ماكفيرلاند تيوبس من بيور كولوني انت عازلها، تعمل انكوبيشن في المورر هينتون اجار وتحضن، وبعدين تعمل انتربريتيشن سليم اكوردنج للشركه عشان تعمل دراج اوف تشويس. ده بيساعد في انك تعالج الحاله صح وتهرب من موضوع الانتيبيوتيك ريزيستنس. بس مهم جدا 3 نقاط نكون واخدين بالنا منهم. اول حاجه لازم يبقى لازم كومبليت كورس ما ينفعش يومين 3 هو الكورس 5. ما ينفعش تلعب في الدوز تبقى كل 8 ساعات تعملها كل 12 ساعة. ما ينفعش يبقى 100 مللي تخليهم 50 عشان توفر لصاحب المزرعة أو لوات ايفر اللي هيستعمل الدواء. مهم قوي يبقى كومبليت كورس، مهم قوي يبقى فيكسد انترفالز، مهم قوي الدوز عشان كومبليت سكسسفول تريتمنت ويز انتيبيوتكس. ثانك يو.